Immunodeficiency diseases. These are disorders in which the immune system's ability to fight disease is compromised or entirely absent. Immunodeficiency diseases are broadly categorized into two. Number one, primary immunodeficiencies, and number two, secondary immunodeficiencies. Primary or congenital immunodeficiencies. These are often due to inherited genetic mutations that affect the development or function of the immune system. Primary immunodeficiencies has the following categories. Number one, the B cell deficiencies, which affect the production or function of antibodies. Examples of B cell deficiencies include X-linked agammaglobulinemia, a disorder where the body fails to produce mature B lymphocytes, leading to very low or absent immunoglobulins. It is X-linked recessive, so it mostly affects males. The disorder is caused by the mutation in the Bruton's tyrosine kinase gene on the X chromosome. The second category is the T cell deficiencies, in which the T lymphocytes or the T cells are absent, reduced, or dysfunctional. Examples of T lymphocytes include the CD4 helper T cells, which activate B cells, macrophages, and cytotoxic T cells. The CD8 cytotoxic T cells, which kill infected or abnormal cells, and the regulatory T cells, which suppress immune overreaction. Examples of T cell deficiencies include Wiskott-Aldrich syndrome, which is a rare X-linked recessive primary immunodeficiency disorder that affects multiple components of the immune system, which include the T cells, B cells, and platelets. It is caused by the mutation in the Wiskott-Aldrich syndrome gene, which is critical for cytoskeletal function in blood and immune cells. Example number two, severe combined immunodeficiency, which is a group of rare, life-threatening primary immunodeficiency disorder characterized by a profound deficiency of both the T cells and the B cells, and often the natural killer cells. And therefore, it results in extreme vulnerability to opportunistic infections starting in early infancy. Severe combined immunodeficiency can be caused by different gene mutations, commonly the gamma chain of cytokine receptor and number 2 adenosine deaminase deficiency. Category number 3 of primary immunodeficiencies is phagocytic cell deficiency, where the function or number of phagocytes, especially neutrophils and macrophages, is impaired. So these cells normally engulf and destroy bacteria, fungi, and debris. When defective, it leads to recurrent bacterial and fungal infections. Examples of phagocytic deficiencies include chronic granulomatous disease, which is X-linked recessive, most common, or autosomal recessive forms can also apply, which comes as a result of inherited defect in nicotinamide adenine dinucleotide phosphatase oxidase enzyme that results into no production of reactive oxygen species needed to kill ingested microbes. In this case, phagocytes can engulf microbes but cannot kill them, which can lead to chronic inflammation and granuloma formation. Second example of phagocytic deficiency, leukocyte adhesion deficiency, in which there is defect in CD18 integrin on neutrophils, and this impairs migration of neutrophils to infection sites. The fourth category of primary immunodeficiency is complement deficiencies, which are immune disorders caused by the absence or malfunction of one or more complement proteins, which are essential components of the innate immunity. So the complement system helps destroy pathogens, enhances phagocytosis, and supports inflammation. And the complement system consists of over 30 proteins that, number one, opsonize or mark pathogens, for example, the C3b. Number two, recruit inflammatory cells, for example, complement 5A, and lyse microbes via the membrane attack complex, for example, C5b.